Okay, welcome to this Crossrail Archaeology update. For some 5 years we've been excavating sites right across the city. You may remember last summer we were excavating in Liverpool Street, and in particular the burial ground at Bedlam. Now, one of the major discoveries there was a mass grave which we suspected came from 1665. And part of the analysis that's been going on over the last several months is to understand, if we can, whether those people actually suffered from the Great Plague, 1665 Great Plague, and can we identify the Yersinia pestis, ancient DNA, within those samples. So let's go now to Mike Henderson over at the MOLA labs, who's going to explain exactly what we found.
The Great Plague was responsible for the deaths of 100,000 people, nearly a quarter of London's population. We know that the new churchyard was used for plague burials. People like Defoe, who described mass burial pits, but people thrown in haphazardly and naked. The evidence from the new churchyard suggests that people were actually placed in coffins in orderly rows, utilising all available space. So the degree of care and attention was still given to them. The pit was half a metre below the surface, and this goes against what we know from rules that were imposed at the time that stipulated that burials had to be at least 6 feet below ground. So this really shows that people were utilising all available space to bury these dead. But at the same time, at a time of mass mortality and high deaths, a degree of respect and dignity was still given to the dead. Now, one of the problems we have as osteologists is that a lot of the diseases that people died from in the past, so things like smallpox and influenza, and indeed plague, they kill you so quickly that you don't see traces or marks on the skeleton. So we have to look for other means to be able to conclusively say if these people had indeed died of these diseases. And as part of this project, we were fortunate enough to be involved in a lot of other scientific testing. Ancient DNA is very vulnerable to contamination and suffers from poor preservation. So we laid out the skeletons under controlled conditions, and we took samples of a tooth from each individual. And this is because the teeth are like sealed capsules that preserve this information better than other parts of the skeleton. And this was sent to the Max Planck Institute for analysis. So the really exciting news from the Max Planck Institute was that indeed these 5 individuals had positive evidence of the Yersinia pestis plague bacterium. They were exposed to the disease. And this is the first time that the ancient DNA of the 1665 the Great Plague has been found in this country. The DNA testing was only part of an array of scientific tests. Following this, we're going to be looking to sequence the full genome of the ancient DNA pathogen. And this will enable us to see the evolution of the disease. How does it compare to previous outbreaks, such as the medieval Black Death? And also, how did it enter Europe and Britain? How did it affect people, and what people were likely to succumb to the disease? And other scientific tests, including stable isotopes, will help us learn about where people were born and where they migrated from, possibly what they ate, and the components of their diet. An analysis of the dental calculus might contain food particles, pollutants, air particles. Through the detailed analysis of the skeletons, we've learned a great amount about the individuals that lived and died and were buried in the burial ground. And bringing together all of this information with all the archaeological information, we're going to learn a lot about these people. All of this evidence and all of this archaeological information and all the stories we've learned will be published in a book to be released in 2017.